Welcome to the fifth in our series of carbohydrate lectures. We're gradually filling out our map of carbohydrate metabolism and all of the pathways that are involved. And today we get to focus on this one here. This is the metabolism of glycogen, how it's stored and how it's mobilized. But let's start with some simple background facts about glycogen. First of all, its structure is quite simple. It's just a long chain of glucose molecules linked via alpha-1,4 linkages. There are also branch points, and this means that the molecule has many ends, a bit like a branched tree. This is an advantage because glucose can be mobilized from each of these ends and therefore speed up the process of storage and of mobilization. Glycogen is the major carbohydrate store in animals, and it's found in the cytosol of pretty much all tissues, but in the liver and in the muscle, it's in higher concentrations. The liver particularly stores up to 6% of its wet weight, and the muscle stores generally between 1 and 2% of its weight in muscle glycogen. But here, it's really crucial to realise that the function of these glycogen stores differs between these two tissues, between the liver and the muscle. In the liver, the key job of stored glycogen is to maintain glucose homeostasis. And this becomes crucial in that early postprandial period as your body begins to fast. And usually, if you don't eat at all, you will have depleted all of your liver glycogen within 12 to 18 hours since your last meal. In contrast, in the muscle, glycogen is used as a fuel for glycolysis in the muscle only, and it's depleted through periods of vigorous and prolonged exercise. Now, it's really important to note here that in the muscle, glycogen is not essential for glucose homeostasis. The muscle doesn't mobilize glycogen and release it as glucose into the bloodstream in the way that the liver does. So in effect, it's used as a high-intensity fuel for exercise. The storage and the breakdown of glycogen actually occurs through two separate pathways. The storage of glycogen is called glycogenesis, and the breakdown of glycogen is called glycogenolysis. So let's have a closer look at these two pathways and see how they differ. Firstly, here's the pathway for glycogenesis. We start with glucose 6-phosphate and we finish with stored glycogen. And there are a series of crucial steps that make all of this happen. This first step, catalyzed by phosphoglucomutase, simply rearranges that glucose molecule so that the phosphate group is attached to the first carbon rather than the sixth. UDP glucose pyrophosphorylase then activates glucose, and glycogen synthesis then adds it to a growing chain of alpha-1,4 glucose residues stored as glycogen. Now, this is a crucial step, the one catalyzed by glycogen synthase. This is the slowest step in this sequence of events, and therefore we call it the rate-limiting step. In effect, if you want to control the pace of any particular pathway, you do so through the slowest step. And the last step is catalyzed by the branching enzyme, which simply rearranges the molecule and puts in place these alpha-1,6 linkages or branch points within the growing chain of glucose residues. You can see how these enzymes function in this picture here. Firstly, glycogen synthase is adding glucose residues to the two ends of the chain of glycogen. And you can see that they extend here and here as that molecule grows. And then, when it gets long enough, the branching enzyme comes along and snips it here, reattaching that chain over here, as you can see in this last picture, via one of those alpha-1,6 glycosidic bonds. So all of a sudden, we've got three ends to our branched glycogen structure. As I said before, this is quite crucial. Each of these ends represent active sites whereby glucose molecules can be attached or mobilised again, which speeds up the process of storage and mobilisation. Let's next look at the process of mobilising glycogen, which happens through the glycogenolysis pathway. In this case, there's only 3 steps involved. Firstly, the branching points are removed through this enzyme combination here. And then glycogen phosphorylase removes 1 glucose residue at a time from each of the ends of the glycogen molecule branches, releasing glucose 1-phosphate. This is then mutated back into glucose 6-phosphate so that it can either be used through glycolysis or, in the case of the liver, released into the bloodstream to maintain glucose homeostasis. I've also included a small diagram down the bottom here to show you how the glucan transferase and debranching enzymes work. Once these chains of glucose residues get short enough, the transferase enzyme shifts 3 of those glucose residues onto the end like this, and then the debranching enzyme snips off that branch point glucose. And then glycogen phosphorylase can keep removing these glucose residues, 1 glucose at a time. There is one nice advantage of using glycogen to provide the fuel for glycolysis. In previous lectures, I've described the anaerobic ATP production from 1 molecule of glucose, and you might recall the magic number was 2 ATP per molecule of glucose anaerobically. However, when this glucose is being provided from the store of glycogen, you actually yield 3 ATP per molecule of glucose, so therefore a 1 ATP advantage. Now the reason for this is pretty simple. It's because the hexokinase step is not required. So here's glycolysis here, converting glucose 6-phosphate through to lactate. The payoff phase of glycolysis still yields 4 ATP, but the investment phase only requires 1 ATP. And the reason why is that mobilised glycogen produces glucose 6-phosphate, so you don't need that extra ATP to provide that phosphate group for glucose. And therefore, the net ATP per molecule of glucose when sourced from glycogen is 4 minus 1 equals 3 ATP per molecule of glucose. Okay, now that's the basics about glycogen storage and mobilisation. But the real question is, how do we regulate those pathways? And in particular, given that they go in opposite directions, how do we reciprocally regulate them? Well, I alluded to this before, but the 2 pathways both have rate-limiting steps. For glycogen breakdown, the slowest step is catalysed by glycogen phosphorylase. And for glycogen synthesis, the slowest step is catalysed by glycogen synthase. So regulation occurs through these 2 enzymes. And you can sort of already guess, if we were going to switch on storage by activating glycogen synthase, then we would want to simultaneously switch off glycogen mobilisation and therefore inhibit glycogen phosphorylase, and vice versa if we were mobilising glycogen. So keep an eye out for that theme when you look at these pathways. Now, the first thing to tell you is that this regulation, this reciprocal regulation, is coordinated through this enzyme here, cyclic AMP-dependent protein kinase. Now, I've definitely talked about this one before. We looked at how it reciprocally regulates glycolysis and gluconeogenesis. And you're about to see that it's got the same job for reciprocally regulating glycogen storage and glycogen mobilisation. Now, when you're starving or when you're stressed, this enzyme is activated via glucagon or adrenaline if you're stressed. They attach to their receptors activated enolate cyclase that produces this second messenger, cyclic AMP, which stimulates the protein kinase, which is quite non-specific. So this enzyme, this cyclic AMP dependent protein kinase, will go around and phosphorylate other proteins that it finds. Now, given that we're starving or that we're acutely stressed, we need to be mobilizing glycogen. So let's have a look at how this occurs. The protein kinase phosphorylates and activates an enzyme called phosphorylase kinase. This enzyme then phosphorylates and activates glycogen phosphorylase, which then merrily goes ahead and starts breaking down glycogen, releasing glucose. So this is great because we've got glycogen being mobilized. The other thing to just spot here, you'll note the key. If it's green, it means that it's in a stimulated form. Now, if we're mobilizing glycogen, then we don't want to be storing it at the same time. So we need to switch off the glycogen storage enzyme. Unfortunately, this controlling enzyme here, cyclic AMP-dependent protein kinase, phosphorylates glycogen synthase, switching it off. You can see I've colour-coded it red. So that's great. We've got mobilisation of glycogen and we've switched off storage of glycogen. Reciprocal regulation. Now at this point, there's another enzyme that I need to tell you about. It's called protein phosphatase 1. It's a phosphatase and it works on proteins, so it's fairly non-specific. And you can see that if it was left to do its merry thing, would go and remove these phosphate groups here, here, and here, effectively removing that phosphorylating signal that's running through your cell. Now, if we want to mobilise glycogen and not store it, then we don't want this protein phosphatase running around undoing all of our hard work. But the good news is it can't. And that's because our central controlling enzyme, cyclic AMP-dependent protein kinase, goes and phosphorylates and activates an inhibitor protein. It's called inhibitor 1, and it blocks protein phosphatase from doing its thing. And therefore, all of these phosphate groups are left in place. So, in conclusion, glucagon during starvation and adrenaline during stress cause net glycogen breakdown. I now want to tell you about another mechanism that causes glycogen mobilization, and this one is specific to your muscle. It's linked to muscle contraction. And when you think about it, this makes quite good sense. In muscle, glycogen is crucial for providing sprinting fuel. This has helped us to survive through years of evolution. You see a lion and you start sprinting and your muscle can directly mobilize its stored glycogen, purely because it's contracting. Adrenaline is great, but in this instance, it'll take just a bit too long to circulate around in your bloodstream and get that mobilization of glycogen happening quick enough. So this is a super fast activation method. When your muscles contract, they release calcium. So during contraction, you'll see these small transient changes in intracellular calcium concentration. Now this has a very direct effect. There's a calcium-responsive calmodulin component to phosphorylase kinase. This will go and phosphorylate and therefore activate glycogen phosphorylase, breaking down and mobilizing glycogen. There's also another calcium-dependent protein kinase that, when calcium levels are high, will be activated and phosphorylate glycogen synthase, switching it off. So therefore, muscle contraction, leading to increased calcium concentrations, activates glycogen mobilization and switches off glycogen storage. Perfect, we've got reciprocal regulation. And this one is super fast, linked directly to muscle contraction itself. It's a bit like giving muscle contraction itself the keys to its own fuel tank. So in summary, muscle contraction causes net glycogen breakdown. Alright, so that was the mobilisation story. Now, let's go and have a look at glycogen storage. As you can imagine, this occurs when you're in a fed state. And so the hormone that's driving the overall action here is insulin, which is released in response to rising blood glucose concentrations. So logically, insulin should cause the storage of glycogen. Let's have a look at how it does this. When insulin binds to its receptor, the first thing it does is it activates phosphodiesterase. This enzyme breaks down cyclic AMP, which of course is the second messenger stimulating cyclic AMP-dependent protein kinase. So in effect, insulin has removed the stimulus that would otherwise have been accelerating this enzyme. So, by removing the second messenger, it's no longer stimulating cyclic AMP-dependent protein kinase. Therefore, we are no longer activating phosphorylase kinase, and in turn, we are no longer activating glycogen phosphorylase. So this enzyme is no longer being stimulated. In addition to that, because our cyclic AMP-dependent protein kinase is not working, it's not phosphorylating glycogen synthase anymore. So it's no longer trying to switch it off. So therefore, we are no longer stimulating the mobilisation of glycogen, and we are no longer inhibiting the storage of glycogen. Now, do you remember this enzyme here, protein phosphatase 1? This was previously being inhibited by an inhibitor protein that had been activated by cyclic AMP-dependent protein kinase. But this guy is no longer active. Therefore, it's no longer activating the inhibitor protein, and therefore, there is no longer any inhibition of protein phosphatase 1. And this means that it can go around removing these phosphate groups here, here, and here. And so this in turn switches glycogen synthase back on so that we're storing glycogen, and it also switches off glycogen phosphorylase and phosphorylase kinase so that we are no longer mobilising glycogen. So that's perfect. We've got storage and we've switched off mobilisation. Again, reciprocal regulation. Insulin also has other more direct effects. It stimulates an insulin-sensitive protein phosphatase, which itself can help to remove these phosphate groups, activating glycogen synthase, and therefore the storage of glycogen. But in addition to this, insulin, because it's stimulated GLUT4 transporters, increases the intracellular concentration of glucose 6-phosphate. Now this substrate here allosterically stimulates the inactive form of glycogen synthase. So even if this enzyme had been switched off, it would still maintain a certain level of activity storing glycogen. In a way, this is a form of feed-forward stimulation. So in conclusion, all of this amounts to insulin causing net glycogen synthesis. Now, of course, glycogen is crucial for glucose homeostasis. So let's revisit our glucose homeostasis scenarios. First of all, there's the short-term one, or the just-fed scenario. In this case, there's lots of excess glucose floating around, which is therefore stored as glycogen. And once your glycogen tanks are full, the excess carbon is then converted into fat. This is all driven via the hormone insulin. As time progresses after your last meal, you'll enter into a short-term shortage phase. In this case, the emphasis is on the mobilisation of glycogen stores, particularly in your liver, and this is driven by the hormone glucagon. We then enter into a medium-term starvation scenario. So here I'm talking about 1 to 2 days and onwards after your last meal. Liver glycogen is largely depleted, and therefore your body starts to put emphasis on glucose sparing. It switches on gluconeogenesis to provide substrate for glucose-dependent tissues, and this, of course, is driven by glucagon. Now, this scenario will go on until you eventually have a feed. Otherwise, you will just starve further. And then the last scenario is the high demand scenario that's seen during stress or exercise. So the emphasis here is on maximising glucose availability, and therefore it's mobilising those glycogen stores. And this is driven by adrenaline. So we can characterise the biochemical and the physiological actions of these main hormones that are controlling fuel metabolism. In the case of insulin, GLUT4 transporters migrate to the cell membranes in both muscle and fat, actively absorbing glucose out of the bloodstream. Glycolysis is stimulated, as is glycogen synthesis, and gluconeogenesis, so the production of glucose in the liver, is actually switched off. In addition, and we haven't discussed this much yet, fat synthesis ramps up, as does protein synthesis. So at an overall physiological level, insulin signals the fed state, acting to lower blood glucose concentrations and increasing the rate of fuel storage. In contrast, the starvation hormone glucagon largely does the opposite of this. It stimulates the production of cyclic AMP in liver and in fat tissue. This reduces the rate of glycolysis, reduces glycogen synthesis, and activates glycogenolysis. It also activates gluconeogenesis, and it activates fat breakdown and protein degradation. And we'll be talking about these last two in the lectures to come. So at a physiological level, glucagon acts to increase glucose release from the liver, aiming to increase blood glucose concentrations and mobilise fuel supplies. And lastly, there's our fight or flight hormone, adrenaline, whose action tends to be more acute. This hormone increases cyclic AMP in liver, muscle and fat tissue, reducing glycogen synthesis, increasing glycogen breakdown, and also mobilising fat. And at a physiological level, this ends up increasing glucose release from the liver and increasing blood glucose concentrations. So, in summary, in this lecture, we've looked at the pathways of glycogenesis, or the storage of glycogen, and glycogenolysis, or the breakdown of glycogen. And we've also looked in detail at how we coordinate the regulation of these two pathways so that they aren't both simultaneously active. If we switch one on, we have to switch the other off. And this, to a fair extent, is controlled through cyclic AMP and the enzyme that it stimulates, cyclic AMP-dependent protein kinase. And just to help focus your studies a bit, I've listed 5 questions that hopefully after listening to this lecture, you should be able to roll through and answer with some level of confidence.